Hello, very good morning to all of you. I, Professor Murvi Rawal, on behalf of Faculty of Pharmacy, Dr. Subhash Technical Campus, welcome you all on this webinar of lung-targeted drug delivery of antitubercular drugs by Dr. Sujit Kumar Devanath, sir. I heartily welcome you, sir, on this wonderful session that you have joined in, sir. All the participants are instructed to switch off their mics during the session and do write your questions in the chat box. Those answers will be, your questions will be answered by sir at the end of this session. Over to you, sir.
Thank you, Urvi. Thank you very much for this introduction. And good morning, everybody. My name is Dr. Sujit Kumar Devnath, and I am a postdoctoral fellow of Department of Bioscience and Bioengineering. And Urvi, just share the screen, please.
Yes, sir. Just a minute, sir.
Yeah.
Just a minute, sir.
Yeah, sure.
Is it visible, sir?
Yeah, visible from my side.
Okay, sir.
Shall I start?
Yes, sir. Sure, sir.
Okay. Thank you, Morbi.
Yes,
And
sir.
good morning, everybody. And my name is Dr. Sujit Kumar Devnath, and I am a postdoctoral fellow of Department of Bioscience and Bioengineering, Indian Institute of Technology, Bombay. It is a short form IIT Bombay. I am heartily welcome you all in this today's webinar, and I am going to discuss on the lungs targeted drug delivery of antitubercular drugs. Next slide, please. Morbi, next slide.
Yes, sir. Is it visible, sir?
No, not Kemnam.
Okay, sir. Just a minute.
Yeah. Yes, came now. Okay, thanks. Before I initiate, I would like to introduce this workplace education and the research of IIT Bombay because that is so much essential for you peoples. And I just want to give in a brief introduction for it. First of all, the first IIT it is a set up in 1951 in Kharagpur. Now the total number of IITs all over the India is stand at 23, and our IIT Bombay it is ranked. It is the first in India according to the QS World University Ranking and 49th in globally. So this IIT Bombay having different departments is having total 15 departments and 20 multidisciplinary centers, one school of excellence and four interdisciplinary programs. All these programs and the functions of IIT Bombay are governed under the supervision of Professor Subhasis Chaudhary, the director of IIT Bombay. And we, IIT Bombay, all the faculties, staffs, and students are really thankful to him to bring this IIT Bombay in number one position in India in terms of education, in terms of the research. This IIT Bombay, that is a completely greenery campus and situated the beside of IIT Powai Lake. Out of this 15 department, the most important one is Department of Bioscience and Bioengineering, and currently we are running two different programs, that is in the MTech program, that is the MSc Biotechnology and MTech Biomedical Engineering. Additionally, some of the students they are converting their MTech program into the PhD program, so they are also getting the dual degree PhD program, dual degree courses, and some students they are directly taking admission in the PhD program also. So we are having this is the four different programs. If you are enthusiastic about the research and development, and I will encourage you all to join the IIT Bombay, especially for the pharmacy students and the professionals also. They can also work and study in IIT Bombay. This bioscience bioengineering department, the right hand side the photo, it is running under the supervision of Professor Rohit Srivastava, head bioscience or bioengineering department. Currently, I am working under him. This VSV having a sixth floor building, and just in this picture, I am just giving the floor of third to sixth floor, and I am working on the fifth floor. The name of the lab is Nanovirus Lab. Next slide, please. Next slide. Now I want to introduce our research area where I am working currently in the Nanovirus Lab. There is a multidisciplinary area it is working here. Some of the group of the people they are working on the cancer biology, some of the people are working on orthopedics, some on in biomems, biosensor, and particularly me and some of the fellow they are working on the drug delivery system. Our lab has developed several devices. You can see in the picture below. These are the several devices that has been developed by our lab, and many of them are commercialized available. Next slide. Now, the today topic is tuberculosis. Before we discuss about the tuberculosis, first we should have to know what are the death-causing diseases that is having a global concern about the WHO. So in that chart, you can find there are different types of death-causing diseases. It will be there. It can be the communicable disease. It can be the non-communicable disease. It may be the injury also. The injury in terms of the road injury or the self-harm. We are talking about the tuberculosis. This tuberculosis already been listed within the 10 highest death causes diseases. You can see when we are ranking these diseases in 2007, it falls the seventh position. But in the case of the 2017 rank of view, it is hike up to sixth position. This hike is near to 11.3%. Now let's see what who is telling. Who's tell the 1.4 million people die from TB in 2019, including more than 2 million people with HIV. Okay, and they are also telling that the one of the disease this is mainly happened only due to one single infections agent, that is Mycobacterium tuberculosis. Next slide. Now, if we are thinking about or watching this types of scenario of 2019, we can observe the globally the name of the tuberculosis is not being displayed in 10 list. But when we are thinking about the low income countries like India, you can highlight it this tuberculosis it has been present in the 8th serial number. It means this disease are mainly occurred for the poor people those are present in these low income countries. This tuberculosis, you need amount of the drugs, and you have to take frequent hospital consultancy. It is not been possible for the poor people to do and check and take regular medicine. So that's why the disease rate and the death rate, it will be much high in these low income countries like India. Next slide. If you're talking about the tuberculosis, this tuberculosis may be two types. One is the pulmonary types, another is the extrapulmonary types. The pulmonary type means mainly the disease is occur in the lungs or different part of the lungs, because this disease, it will be airborne disease. If some people they inhale this microorganism, easily this will be deposited in the pulmonary, means in the lungs, and causing the several diseases. It is associated diseases. You can see these pulmonary diseases can be several types. It can be pneumonia, you can be pleurisy, it can be malaria, cavitary or laryngeal. Similarly, extrapulmonary tuberculosis also be occur in some other part of the body, maybe lymph node, maybe petechiasis, maybe osteal. Mainly, we are mainly focusing here the pulmonary tuberculosis because the occurrence of this tuberculosis is such high. Next slide, please. Here we can see there is a very list of drugs it will be available to treat this tuberculosis. According to the WHO, it will be categorized as group 1 to 5. But depending upon their efficacy, some of the group it will be promoted and some of the group that will be demoted in this 2016 list. We also know in case of the tuberculosis, one single drug cannot be worked out. For that, we give multiple drugs. So, based upon this combination therapy, some of the drugs it will be promoted and some of the drug it will be demoted. So, if it is going on still now, and we can also observe similar rearrangement of these drug classes in future also. Next slide. Now the question is arise. Suppose if we are given one drug, that is the anti-tubercular drug in the oral route, can it be possible for whole the drug it is reaching to the lungs? For my prospect, it is no, because when we are giving the drug to the oral route, only some fraction of the drug that is reaching to the lungs, because most of the drug that will be distributed throughout the body. But in case of the tuberculosis, particularly for the pulmonary tuberculosis, lungs is mainly affected. So if we are giving in the oral route, we cannot achieve the higher concentration of the drug in the lungs. Similarly, if you are giving one drug in the oral route, what will happen? The peak plasma concentration of the drug suddenly goes high in plasma. So what is happened? It gives the toxic effect, maybe for the systemic toxicity, maybe for the organ toxicity. Additionally, in case of the tuberculosis, we are used to give many more combination of drugs at the high dose. What is the result? It gives drug-induced liver injury. So obviously, we have to think about some alternative drug delivery system where we can giving the drug directly to the lungs. For that instance, we are having two delivery route. One is the nasal route, another one is the pulmonary route. When we are giving in the nasal route, what will happen? The drug it is reaching to the lungs. It is true, but some of the drug that is penetrate to the blood-brain barrier and come to the brain also. So that is not being desirable here. So that's why generally we are preferring the pulmonary route. In the pulmonary route, we may give in the drug in the form of the DPI, in the form of the MDI. DPI stands for dry powder inhaler and MDI stands for metered dose inhaler. We know very well if we miss the dose, particularly in the case of the tuberculosis, there will be the chances of drug resistance. And once the drug resistance is happened, you cannot treat, or the treatment it will be very much difficult. For that instant, if we are formulating the nanoparticulate form, then what will happen? You have to take this dose only once in a day. So we can reduce the misdose concept here. Additionally, as I mentioned before, in case of the tuberculosis, we have to giving several drugs, several dose, multiple dose. But if you are formulating the nanoparticulate drug, then you are giving only one dose, and for that 24 hours, the drug it will be remain in the lungs and maintain the lungs concentration of the drug. Next slide, please. So for this formulation purpose, in the next couple of slides, I will guide you how to do this formulation step by step and what are the things that you should have to keep in mind during formulation. The first thing is FTR analysis. What we are doing in the FTR, we are just checking the compatibility issue between the drug and polymer or other ingredients. Whenever we are using the drug into the form of the any dosage form, we are generally giving several chemical reaction. We are giving several additives. So some of the cases, what will happen? There will be the chances of the drug-drug interaction or drug-material interaction. Is there any interaction it is happened or not? That we are confirming from this FTR analysis. Here we perform and scan this FTR of the erythromycin single and in the mixture, and we have estimated whatever the principal peak of the erythromycin and compare this peak with the data from the mixture, and we observe the principal peak remain present in the form of the mixture. So we can assume that this drug, it is the erythromycin, it will compatible with the other ingredients whatever we have required to prepare the formulation. Next slide. This is the materials methods that we are adapting for formulation of this nanoparticle. We have adapted the emulsion solvent evaporation technique. In that technique, we have taken two different solutions. One is the organic phase, another one is the aqueous phase. This organic phase, it is contain drug and polymer, and the aqueous phase, it contains surfactant. We have used PVA, polyvinyl alcohol, as a surfactant. Then what we did? We have added this organic phase dropwise into the aqueous phase. Next, we have sonicated by using probe sonicator to formulate into a nano emulsion. Afterward, this organic phase we are gradually evaporating under magnetic stirrer at room temperature. What will happen? The all the organic phase it will be evaporate. Next, it will form the nano suspension. Afterward, we have centrifuged it with high RPM to settle down all the nanoparticles at the bottom. Next, we have taken the supernatant. In this supernatant, we have calculated the drug content that is not been entrapped into the nanoparticle by using HPLC or UV spectrophotometry. Whatever the sediment we got, that we perform the freeze drying in presence of mannitol 2% W by V. After getting this freeze dried product, we have again characterized in the different way. Again, we are performing FTR, we are performing DSC. Some of the sample we have kept in the stability chamber for studying the stability study. We also perform the morphology of the structure by using same. We have characterized by using the zeta size, it is a particle size. Next slide, please. So in this process optimization, first process optimization is the drug PLGA ratio optimization. So what we did, we have taken the different concentration, different amount of PLGA. With this varying PLGA, we have prepared this formulation as the method we have described previously, but we have kept some of the parameter constant. As for example, drug amount we have kept 50 mg, it will be constant for all the formulation. PBA concentration, that is 0.25%, it is remain constant. Organic phase volume, that is a 4 ml, it is remain constant. And aqueous phase, that is a 10 ml. Next, we have checked what is the influence of this changing this PLGA amount on the other factors, that is the Z average value, PDI, zeta potential, and percentage drug entrapment, that is in the short form, it is PDI. Here, Z average value, it is giving the average value of the particle size obtained from the instrument zeta sizer. PDI, it is known as polydispersibility index, which gives us the idea how segregated or how distributed our particle. Is there our particle, it will be in the narrow range distribution or it is preparing the different size particle. Zeta potential, it is giving an idea what will be the charges of the particle on the surface. The value, it is give us the idea that the particle it will be aggregate or not. If the size, if this charge it is lies within plus minus 5, there will be the chances of aggregation. So what is our expectation? Our expectation, it will should be the value should be more than plus minus 5. It may be plus 5, it may be minus 5. Beyond that, when this value it will be reach or exceed the 30, we can assure you that the particle it will be stable, there will not be any type of aggregation. So what we observed that if we increasing the polymer amount, the particle size is gradually increasing. Simultaneously, the percentage drug entrapment also increase and the charges it will be varies. So what we have to proceed now? We have taken the such formulation where the aggregation of chances is so much less. That's why we have taken the fourth formulation, whatever it is highlighted with the blue ink. Next, next, we have kept this formulation all the parameter as it is. Again, we are preparing four different formulation with varying concentration of the PBA. Previously, we are using 0.25%. Now we are gradually increasing the amount of PBA in different formulation. Our observation. Let's see, when we are increasing the surfactant concentration, the particle size it is gradually decreasing. Additionally, when the particle size is decreased, the percentage drug entrapment also decrease simultaneously. So at that time, at what basis we have chosen the best formulation? We have chosen the best formulation based upon its percentage drug entrapment, whatever we have achieved at 0.5. Additionally, we can observe the zeta potential also it will be higher than the previous formulation, that is 5.7, it is now it is a 8.1. So we have optimized this formulation based on this surfactant concentration. It is taken as 0.5% WV of PPA. Next, again we are further modifying this formulation. Next slide, please. Now you can see for the organic phase volume, we have taken two organic phase, two solvent system. One is DCl, another is acetone. Before we are formulating, we have checked our drug and the polymer, it will be dissolved in which solvent, which organic solvent. At that time, we observed when we are taking the combination of DCM and acetone, the drug and polymer, it will be dissolved quite easily. Now, we have increased the amount of acetone. Previously, we are using 1.5 ml. Now we are increasing the amount of acetone by keeping the DCM value same. Next, after formulating all these parameters, we check that the parameters like Z average value, PDI, zeta potential, and the percentage drug entrainment, the value is significant changes. Means what? When you are increasing or diluting the organic phase concentration, the particle size it will be decreased. If the particle size it will be decreased, then the drug entrainment also be decreased. Here you can see the first graph. When we are increasing the acetone amount, means your organic phase volume also be increased. The particle size it will be decreased. But in the last cases, again the particle size increase. Why? Because when the smaller particle it will be achieved, but the zeta potential it will be slow down. Then what will happen? Again, there will be the chances of the particle aggregation. Due to this particle aggregation only, the last cases the particle size increase. So that's why we observe that if we are increasing the acetone amount also, we are not getting a better result. So that's why we have further modified this formulation. Next slide, please. We have observed the same analysis of this particle, and why we are performing this same analysis. Generally we have to check the particle morphology and shape and size. You can see when we are performing it in 19.99 kx, the particles it will be shown as very small, and most of the cases all are the particle are spherical in nature. So whatever the particle we are prepared here, these are all spherical particles. Next slide. Now we have to check this differential scanning colorimetry of the drugs as well as the particles. This study generally we are performing to check is there any degradation of the drug or not. So for that first we have run this pure ethionamide. When we have run this pure ethionamide, you can see the red dot. Here you can see we are getting one sharp peak. When we are getting the sharp peak, it means this drug it is in the crystalline form. Due to this crystalline form only, this endothermic means the peak it is coming downward. This endothermic peak it is came as sharp. We also check this melting point of this PLGA 5050 also. This PLGA 5050 it is melt near to 60 to 63 degrees centigrade. Here also you can see this green line. This is representing the PLGA 5050, and it is also not much sharpness as like of ethionamide, but it is also a sharp. Okay, but when we are formulating this nanoparticle of erythromycin PLGA, we can observe that both the peak that is in the 60 degrees centigrade as well as the 160 degrees centigrade for the ethionamide, both the peaks are present. It means once you are formulating the nanoparticle, there will be the nothing changes in the melting point of the drug as well as the PLGA. But we have observed the peak it will be little bit broadened. This broadening that is mainly due to the conversion from the crystalline particle to the amorphous particle. The shape has been changed. Okay, the shape change it is not been an issue. There should not be significant shifting of melting point that we should have to keep in mind. Next slide, please. As we discussed, we have used two different solvent. One is DCM, another is the acetone. But we have to also know, according to the ICH guideline, we have to perform the residual solvent analysis. This residual solvent can be classified in three groups. class 1, class 2, and class 3. This class 1 group, this demonstrated highly human carcinogenic and also having environmental hazards. So we should have to restrict significantly this class 1 solvent in our formulation. Class 2 solvent, it is giving irreversible or reversible toxicity. So we have to restrict class 2 because we cannot take it as a huge quantity. For every solvent fall under the class 2 having one permitted daily exposure value that we should have to check. And the class 3 is low toxic potential drugs or solvent. This low toxic potential means whatever the solvent it will be fall under this category, it's having one fixed value, that is the 60 mg per day. You have to take 60 mg, less than 60 mg, not more than that. Our acetone, it is fall in the class 3 category, and DCM, it is fall in the class 2 category. So that's why we have to check in our final formulation what is the trace amount of this solvent it will be present. So that's why we have performed the residual solvent study using Headspace GC. So first we have run the standard acetone, next we have run the standard DCM, and next we have run our formulation. And we observe that the DCM it is not been available, not been present in our final formulation, but the acetone it will be present at the concentration of 0.65 ppm, but this amount is quite less rather than the PDE, that is the permitted daily exposure. So our formulation it will be safe for human use. Next slide. As you know, this nanoparticles flow property it is not been good. So obviously we have to increase the flow property. For increasing the flow property, we have to add lactose. As this is the formulation it will be giving to the pulmonary route, we cannot use a simple lactose. We need a special type of lactose, that is the inhalable grade lactose. This lactose I have received from Kerry Groups USA as a gift sample. So what we did, we have added this inhalable grade lactose with our nano formulation, nano freeze-dried particles, and observe the angle of repose, Car's index, Hausner ratio. Because this Car's index, Hausner ratio, angle of repose having one fixed value. From the value we can detect as our formulation flow property. It will be good, bad, worse, very poor. What is this? So we have to improve this flow property, and we have checked that when we are using at 1:1 ratio, the flow property it will giving the optimum. This is good. Next slide. Not only that, after doing this DPI formulation also, we have to verify as this formulation it will reaching to the lungs or not. For this testing, what we perform, we have performed this Anderson cascade impactor for checking the MMAD value. MMAD value means mean median aerodynamic diameter. This cascade impactor having 8-stage filter. What we perform, we have passing our powder using these devices, and we have checked what is the amount of material that is deposited in every plate, and we have calculated how much quantity that will be deposited in every plate. This value we have directly put into this below mentioned website calculator, that is the mmadcalculator.com Anderson. Here we have put this value and we are getting this MMAD value. This MMAD value highly significant for administration or delivering of the dry powder inhaler to the lungs. If the value lies between 1 to 5, it can signify the material can be delivered directly to the lungs. If the values is less than 1, means the drug or the DPI formulation, whatever we have inhaled, it will be expelled easily. And when the value is goes beyond the 5 micrometer, it is deposited on the thoracic part. It is never reached to the lungs. Here we can see the MMAD value that is come 1.79, means this formulation can reach deeper to the lungs. Next slide, please. Now we have observed the release pattern of our formulation. We have performed the 24 hours release study. After the release study, we have plotted into a different model, zero order, Higuchi, PIPAS model, and each model we have calculated the R square value, and we observed the R square value for zero order release profile is higher rather than the Higuchi model as well as the PIPAS model. So we can say that this formulation it is followed the zero order kinetics. For the estimation purpose, this ethionamide, we have developed one different methods in the UV spectrophotometry method. Using this method, we can successfully estimate the drug content in our nano formulation, in the marketed formulation. That's why we can assume whatever the development method we have developed, it is also be validated according to the ICH guideline also. And the same things we have published in International Journal of Drug Development and Research. Next slide. Now we have performed the stability studies of our final DPI formulation for 6 months. During this 6 months, we have maintained the temperature 25 degree plus minus 2 degree centigrade and the relative humidity of 65% plus minus 5% RH. We have observed there is not any significant changes in any terms. In case of the PDI, zeta potential, PDE, percentage drug release, but we have observed slight hike in particle size with time. But this is a nominal changes that will be happen for every formulation. So we can assume that our formulation it will be stable in accelerated stability study for 6 months. Next slide. Again, the main problem for delivering of this DPI. For this purpose, this main Milano, Italy, they have given this monodose inhaler and nasal insufflator as a gift sample. But these devices mainly helpful for delivering of the drugs for the human use, not for the animal purpose. That's why we fabricated one different devices which will be helpful for delivering the drugs directly to the trachea region of the animal. We have taken this needle. This needle we are using by the ENT specialist for anesthetize the thoracic part. And we also taken one catheter that is the green color attachment that you can observe in this picture. We have attached both the things and we have used one 5 ml syringe, 2 to 5 ml syringe for generating the air. In the top portion, we have filled our formulation and we have using this air, we have passing the all the content through these nozzles. Here in the IIT Bombay, these devices we have developed by using the 3D printing, but also having the similar action, similar delivery efficiency as we have developed previously. And both the cases, the delivery, it is achieved as 98.25%. Next slide. Now come to the animal experiment, that is the in vivo experiment. For this experiment, we have taken the oyster rat and we have grouped this rat into two different groups. First group that will be treated with our formulation and the second group that is be treated with pure ethionamide. And we have taken the 6 point where we have to withdraw the samples. For the each point we have taken the three red. So that's why our requirement was 36. And at each time point we have sacrificed 3 red and we have estimated the drug concentration in plasma as well as in lungs. Before we are administering the formulation, we have anesthetized all the rats by using ketamine and xylazine. For administration, you can see in the picture, we have taken one forceps and sprayed the mouth of the rat, and we have taken the pull out this tongue outside, and at that time you can see the trachea region of the rat. In the trachea region, we have insert our needles and spray the powder formulation. Next slide. As it told, we have observed the lungs distribution as well as the plasma distribution of the formulation. In both the cases, we have administered the plain ethionamide as well as the nanoparticle loaded ethionamide. We observe when you feel giving in the free form, not in the nanoparticulate form, the sudden height of ethionamide, it will be high and it is reached 4.62 microgram per ml in 1 hour, and rapidly it will be eliminating from the lungs. We can detect this ethionamide concentration at the 6 hour, but it is not been detected at 12 hours and the 24 hours. Means it will be eliminating from the lungs after the 6 hours. Whereas when we are giving this ethionamide in the form of the nanoparticle, this ethionamide is present in the lungs for 24 hours. But you can see the last value, it is in the 24 hours, it is a 0.39 microgram per ml. It is less below the minimum inhibitory concentration of the ethionamide. But our achievement is this formulation, it will be persist in lungs for 24 hours that we achieve. Apart from this lungs distribution, we also observe the plasma distribution because we are giving the drugs in the lungs, not in the plasma, but also it will be distributed in the plasma. Our aim is we have to restrict the drug concentration in the lungs. We have to restrict the distribution in the plasma. What we observe when we are giving in the free form, the drug concentration it is 0.36, but again after 12 hours it is not been detected at all in the plasma, but the plasma concentration when we are giving in the nanoparticulate form, it will be detected. But whatever the concentration we have achieved in plasma, it is quite less rather than the minimum inhibitory concentration of the ethionamide. So we have achieved our goal that whatever the drug that will be restricted in the lungs, it will not be distributed in the plasma. So this is the rest of the pharmacokinetic parameters that we can also observe. When if you are giving in the pure form, the AUC value it will be less. When it is giving in the nanoparticle form, the AUC value is much more high. It signifies that ethionamide in the form of the nanoparticle, it will be stay prolonged period of time in the lungs as well as in the plasma also. And the Tmax we are getting in 1 hour. Next slide. Now come to our conclusion. We have formulating this erythromycin nanoparticle by using this solvent evaporation method, and we have observed they are having the significant influence of this drug PLG ratio, surfactant concentration, and organic phase composition and volume on the performance of this nanoparticle. We have observed The DPI MMAD value is 1.79. It means it will be reaching directly to the lungs. We also observed that in vitro drug release profile giving the initial burst release. Due to this burst release, the concentration of the drug in the desired site it will be high. It will goes above the MIC value, and after that it is followed the zero order kinetics. And the most important thing, whatever the formulation we have prepared, it is maintain the MIC value more than 12 hours with a single dose in the rat. Although the 24 hours value is less than the minimum inventory concentration, but we assume if we are giving this drug in the repeatedly daily basis, the drug concentration in the lungs it will be maintained above MIC. This article we have published in Journal of Pharmaceutical and Biomedical Analysis with an impact factor of 3.915. So I request you all just go through this article, and if you are having any doubt you can mail me. I'll reply you with the appropriate answer. Next slide. Now we have to think about whatever the formulation we are trying to prepare, that is in the preclinical stage. Okay, you can see lots of antitubercular drug that is already been performed in the preclinical stage, but how many number of drug that is reaching to the clinical stage? Only one, that is the capreomycin. Okay, next slide. But in the approved list, not a single antitubercular drug it will be approved for the pulmonary delivery. Recently, we have published one review article, that is in expert review of respiratory medicine, where we have given this type of information. Okay, lots of the people they are working on the pulmonary system, they are performing the preclinical study, but very less number that is reaching to the clinical stage, and not a single formulation that will be passed or approved for pulmonary delivery. Means they are having several lacuna. So I am requesting everyone, those who are having interest in the research, and also encourage, you should also work on the respiratory medicine, that is a pulmonary delivery system, to bring more formulation into the preclinical to the clinical phase, and from the clinical phase to the approved phase. Next slide. Now acknowledgement. First of all, I would like to acknowledge Professor S. Sai Shivam, Principal, Ethionamide Institute of Pharmacy. He is my PhD supervisor. Today I am in the position mainly due to him only. He is always motivative and encouraging person and make me self-dependent. For this, I always be thankful to him. Next, in the right hand side, my PDF supervisor, Professor Rohit Srivastava, HOD, Department of Bioscience and Bioengineering, IIT Bombay, is a great professor. Recently nominated as prestigious Himanshu Patel Chair Professor, and also associated with many governing bodies of Government of India. And I am lucky as I got the opportunity to work under him. Lastly, not the least, I want to acknowledge Dr. Subhash Technical Campus, Junagadh, Gujarat, for inviting me this webinar and arranging this event in well manner. Special thanks goes to Dr. Chintan, Professor Jay, and the faculty members of the pharmacy department. I am wishing all the best to all the students and the attendees of this webinar. Next slide. Thank you for attending this webinars and being with me. If you are having any doubt, any discussion, any collaborative work, you may reach me to Dr. That is a drskdev@gmail.com or drskdev@iitb.ac.in. I also encourage you all to visit my profile and follow in LinkedIn, Google Scholar, ResearchGate, etc. for my research activity and the update. And thank you very much. Over to you, Morvi. Any question from your side, Morvi?
Yes, sir. Thank you so much, sir, for such an interactive session. Now there are some questions, sir, we have received in chat box. I would like you to answer them.
Yeah, go ahead.
Sir, one question is how can we apply this lung targeted drug delivery system on treatment of COVID-19?
Yes, it is a very good question. Actually, we are also still working on this one project with the collaboration of Sign Hospital. We're also trying to develop this formulation, but the main thing is the mutation of this COVID. Okay, if we are trying to prepare any type of formulation, no, it will giving directly to the lungs. It is okay, fine, but actually for particular drug it will not been established yet what we should have to administer for the COVID. Okay, for the symptomatic relief what we have to do means we can giving one mucolytic agent or addition with any antiviral drugs, because most of the cases what we observe, no, the people they are having a pneumonia and having several mucus secretion in the lungs. Due to this they are not able to breathe, so that's why the deficiency of the oxygen it will be occurred, and the people are giving the high level of oxygen supply. So, as a symptomatic relief, currently what we can suggest is we have to giving one any type of antibiotic addition of the mucolytic agent or any type of antiviral for the mucolytic agent to suppress the symptoms. Okay, because if this virus it will be enter into the body, the antibody will be generating automatically due to the immunity system. So we should have to give the sufficient time to the body to work as alone. Okay, as we are not having any particular drugs, okay, that can be 100% cure this COVID. Okay, so I can suggest this combination, that is a mucolytic agent with any types of antibiotic or any types of antiviral drugs for pulmonary administration.
Okay, sir. Sir, next question is which drug is most suitable for such kind of drug delivery system in treatment of TB?
Actually, it is depends upon in which route you want to administer. If it is oral route, it is not a issue. If it is intravenous route or the pulmonary route, then you should have to think about the properties of the drugs. Suppose in some cases, if you are thinking that your drug having the short biological half-life, in such condition you are giving these drugs repeatedly to the oral route to maintain the minimum inhibitory concentration for the prolonged time. So if you are taking in the nanoparticular form, then what will happen? The drug it will be released in a sustained manner, all a predetermined rate to giving the sustained effect for the prolonged period of time. So I generally prefer those drugs for pulmonary administration having a low minimum inhibitory concentration, means in the less concentration it is giving the higher efficacy and the dose requirement is less. Okay, but
Okay,
also
sir.
we should have to think about how we should have to modify the all the drugs and to bring into the pulmonary system, that is the research. Okay, thank you.
Okay, sir. Sir, next question is 3D printing device commercially available in market for drug delivery through lung targeted system?
No, it is not been available. What we did, we have fabricated these devices, and we have applied it on the animal also in collaboration with NRH Mumbai, that is the
Okay, sir.
ICMR lab. Some of our TB project it is going on there. So we have tested it, and we found the efficacy is quite good, but we never think about it commercialization. If the demand it will be more, then we can think about.
Okay, sir. So next question is, as you say, generally in the treatment of TB, we are using combination of drugs. But when we using nanotechnology, can we use any single drug as if for treatment of TB?
It is depends upon the activity. Okay.
Okay,
For
sir.
our case, we have chosen not only one drug, we have chosen the two drugs. One is ethionamide and the prothionamide. But in this presentation, I only display about the ethionamide, not the prothionamide. Preferably, one single drug cannot be success much more in the tuberculosis, but you can administer more than one drugs in the nanoparticle form to pulmonary route. Okay,
Okay, sir.
so in that condition, what you should have to do in the same formulation, you can incorporate multiple drugs. Otherwise, you can prepare the two to three different formulation of two to three different drugs. Next, you have to mix when you have to prepare the DPI formulation with the lactose. Okay, this formulation you can administer. Again, it is study based. You have to check it is been passed through all the testing. That is the MMAD value should be within the 1 to 5. Then only it will be suitable.
Okay, sir. Okay, sir. So I would like to thank you for joining us for this wonderful interactive session. I would also like to thank our participants for joining in and making this wonderful event happening one. This wouldn't be possible without you all.
Right.
And all the participants are hereby instructed to fill their feedback form that they will receive in their mail, and after that they will receive their certificate of participants. Once a minute, sir.
Yeah.
Sir, I would like to present you the certificate of appreciation. Thank you so much, sir, for joining in. We are pleased to
Thank
have
you.
you.
Thank you.
Thank you very much,
Thank
sir.
you, Murthy.
I
Okay.
thank you
Okay.
in behalf of Faculty of Pharmacy. Thank you so much, sir.
Okay. Thank you. Thank you. See you. Bye.
Thank you, sir. Bye, sir.
Okay.